Group 8 practical video, Koch Postulate practical video. This is our group members. Koch Postulate practical 2. The infected group sample was sliced through and any changes in color and texture of the infected tuber were observed. A sterile inoculation loop was used to collect the infected carrot, which was then transferred onto agar plates. It was used to carefully remove a pustule from the infected potato or carrot, and it was placed onto the agar plates to prepare a PDA sample. A sterile scalpel blade was also used to carefully remove a pustule from the infected carrot and potato under the microscope. A drop of cotton blue stain was placed onto the pustule and it was covered with a cover slip. The cover slip was gently pressed down. Observations were recorded. The results observed for carrot and potato are as follows. Koch Postulate 3. The lesion surface was touched using a sterile inoculation loop, and the strict plate method was performed onto an air plate. For subculturing, a small piece of mycelia plug was taken from the edge and placed onto a new PDA plate with the mycelia facing the surface of the agar media. The inoculated PDA and NA plates were sealed using parafilm and incubated at 25 Celsius for 24 to 72 hours. Koch Postulate Practical 4. The work area was wiped with 70% ethanol. The carrot was peeled and sliced into this. The healthy carrot was soaked in 1% sodium hypochlorite for 15 seconds, rinsed with water, and the surface was sterile by wiping with 70% ethanol. The carrot slices were rinsed twice with sterile distilled water and placed onto sterile filter paper to remove excess water. A sterilized inoculation loop was used to pick up a single colony from the pure culture, and the bacteria was smeared on the surface of the carrot disc. The healthy potato was soaked in 1% sodium hypochlorite for 15 seconds, rinsed with water, and the surface was sterilized by wiping with 70% ethanol. A scalpel and inoculation needle were flamed using an alcohol burner and allowed to cool in the air for 10 to 20 seconds. A sterilized inoculation needle was used to cut a small piece of agar with the fungus from the pure culture. A healthy potato tuber was inoculated with the microorganism by inserting the small piece of agar with the fungus into it. The results were as follows. The inoculation loop was sterilized and bacteria from the surface of the infected carrot were smeared onto new agar plates. A small piece of lesion was cut from the infected potato sample using a sterilized knife, sterilized in 1% sodium hypochlorite, and transferred to new PDA agar plates. Koch Postulate Practical 5. A sterilized inoculation loop was used to smear the bacteria on the surface of agar plates. A portion was placed on a slide, a drop of glue stain was added, and it was covered with a cover slip before being observed under the microscope. The steps were repeated to observe the PDA sample. The results observed under the microscope were as follows.